Good morning, students. We are going to start with lesson number 11, Enhancement of Food Production. You know that food is a basic need of human being or all the living organisms. It keeps us alive, strong and healthy. But due to increasing population, the demand for the food is increasing day by day. To meet the increasing demand for food, there is need for improvement of food production, quality wise as well as quantity wise. Plant breeding and animal breeding, that is hybridization in plant as well as in animal, help us to increase the food production. Plant breeding, the improvement in the heredity of crops and the production of new superior varieties of existing crop plants is called plant breeding. You must have heard about hybridization technique. It is a cheap method that offers greater possibilities in the crop improvement than other method. You know that in hybridization, two or more you can say that desirable characters of two or more varieties of the plants are mixed. So that is called as hybridization or hybrid plant or animal. So hybridization can be intravarietal or intervarietal, interspecific or intergeneric. We are going to study now different steps involved in the hybridization technique or in the plant breeding technique. You can see here are some steps given, like collection of genetic variability, evaluation and selection of parents, cross hybridization of selected parents, selection and testing of superior recombinants, testing, release and commercialization. So these are the main steps involved in the plant breeding. Students, this may come to at least the steps involved in the plant breeding for 2 marks. So learn any 4 steps properly. So main step, very first step from the steps, collection of genetic variability. In wild relatives of many crops, pre-existing genetic variability is available. Collection and preservation of wild varieties, species and relatives of the cultivated species is a prerequisite for effective exploitation of natural genes. The entire collection of plants or seeds having all the alleles for all genes in a given crop is called germplasm collection. Germplasm conservation can be done in following ways. In situ conservation, it can be done with the help of forest and natural reserves. Ex situ conservation, it is done through botanical gardens, seed banks, etc. The second step is evaluation and selection of parents. It is an important and essential step. The collected germplasm is evaluated to identify plants and desirable characters. The selected parents must be healthy, vigorous, and should show desirable characters. The selected parents are self for 3 to 4 generations to make them pure or homozygous. Pure lines are created wherever desirable and possible. The third step in the plant breeding process is hybridization. The variety showing maximum desirable features is selected as female parent and the other one is male parent, which looks good characters found in recurrent parent. The pollen grains from anthers of male or desirable male parent are collected and then artificially dusted on stigma of the female flower that is also desirable or emasculated flower. What is meaning of emasculation? Emasculation is nothing but removal of anther with the help of forceps, that we are going to study in the practical. Pollination is followed by seed and fruit formation in due course. The seed thus obtained is called as hybrid seed. There are some steps involved in the hybridization technique: collection of variability from germplasm or gene bank, evaluation and selection of parents, selection of parents plant or parent plants with different qualities, selected parents self for 3 to 4 generations to make them homozygous or true breeding, identification of parents as male parent and female parent, collection of pollen grains from the flower or from the male parent, removal of stamen from the flower of the female parent, that is called as emasculation, artificial cross pollination, that is dusting of pollen grain on the stigma artificially, then bagging and tagging of the emasculated flower of female parent, that is whatever that cross pollination artificially is done, that flower is bagged and then label is tagged on that. Then development of fruit and seed representing F1 generation or F1 hybrid. The next step is selection and testing of F1 hybrid for combination of desirable characters. Then field trials for productivity, and the last is testing and release of variety. So these are the steps which are given on page number 248 of biology textbook. Please learn that flow chart. The next step involved in the plant breeding is selection and testing of superior recombinants. It is crucial to the success of breeding objective and requires careful study, evaluation of progeny. It yields plants that are superior to both parents. That means both the parents are having good quality. These are self-pollinated for several generations till they reach a test of uniformity, so that the characters will not segregate in the progeny. The next step is testing, release and commercialization of new cultivars. The newly selected lines are evaluated for their yield and other agronomic traits of quality, disease resistant, etc. This is done by growing them in research field and recording their performance under ideal fertilizer application, irrigation, that is watering, and other crop management practices. The material is evaluated in comparison to the best available local crop cultivar. Indian hybrid crops, wheat and rice. In 1960, wheat and rice production increased tremendously. Norman Borlaug developed semi-dwarf varieties of wheat, that is Sonalika and Kalyan Sona are two of hybrid wheat varieties grown in India. Semi-dwarf rice varieties were taken from IR8 and Taichung Native 1 from Taiwan and introduced in India. Jaya, Padma and Ratna are the better yielding semi-dwarf rice varieties that are developed later. The next is sugarcane. Here in the nearby picture you can see sugarcane juice which is extracted from the sugarcane. Saccharum barberi and then Saccharum officinarum. These are the varieties of sugarcane. Saccharum officinarum has thicker stem and high sugar content, but it does not grow well in North India. These two varieties were crossed to get the desirable qualities of both. That is, hybridization is done in between these two qualities or these two species. So some of the hybrid varieties of sugarcane are CO419, 421, 453 are high yielding and having high sugar content, are developed in India at Tamil Nadu. The next is millets. Hybrid maize, jowar and bajra have been successfully developed in India. These varieties are high yielding and resistant to water stress. Plant breeding for disease resistant. Some of the diseases caused in plants are, the chart is given on page number 249 of your textbook. So first pathogen is fungi. The disease of plant is brown rust of wheat, red rot of sugarcane, late blight of potato. Then due to bacteria, black rot of crucifers. And then due to viruses, TMV, that is tobacco mosaic virus. So biotechnology, some disease resistant plants are developed like wheat. The variety is Himgiri. This is resistant to leaf and stripe rust, hilbert. Then brassica, Pusa swarnim. This is resistant to white rust. Then cauliflower, Pusa shubra, black rot and curl blight black rot. Then chili, the variety name is Pusa sadabahar, resistant to diseases chili mosaic virus, tobacco mosaic virus and leaf curl. You can see here in the nearby pictures. These are some plant diseases. To cure these plant diseases or to prevent these plant diseases, there are some different varieties of plants are formed, created by hybridization or plant breeding technique. So you can just go through the chart. These are the different varieties of the wheat, brassica, cauliflower, chili are formed biotechnologically or in the hybridization technique to resist the different types of diseases. Plant breeding for disease resistance. Conventional breeding technique, that is, it include mutations, sudden genetic change in plants and screening them for resistance help to identify desirable genes. Such plants can be multiplied directly or can be used in breeding. Other breeding methods are selection among somaclonal variants and genetic engineering. So here in the picture you can see virus resistant plant is formed, which is giving high yield crop. This virus resistant and high yield crop. So both the varieties are combined and giving rise to one plant. So one character is virus resistant and second is high yielding. Here in this third plant you can see both the varieties of both these plants are formed combined together. Plant breeding for developing resistant to insect pest. Development of morphological characters like hairy leaves in cotton and wheat develop vector resistant from jacids and cereal leaf beetle. Beetle leaf means khane ka patta, respectively. Solid stem in wheat leads to resistance to stem borers. Biochemical characters provide resistance to insect and pest. For example, high aspartic acid and low nitrogen and sugar content in maize lead to resistance against stem borers. The nectarless cotton having smooth leaves develop resistance against bollworms. So here are some varieties you can see. Brassica, that is mustard plant. Pusa gaurav, that is a hybrid variety. and insect pest is aphids. Then flat bean, Pusa Sem2, Pusa Sem3, these are hybrid varieties. Then insect pests are jacids, aphids and fruit borers. Then the next is bhindi, that is okra. Variety, hybrid variety is Pusa Savani, Pusa A4. Insect pest is shoot and fruit borer.